So we're going to take a tour of the lab with a little bit of how using the lab and the environment in the lab, it helps with doing research on scleroderma. Feel free to stop me anytime. And some of it is videos, and keep in mind, we did the videos in-house with our phones, so they're a little shaky, but at least it gives you an idea of what it really looks like. All right, so I want to start off with why are we doing research in general? And then I want to try and emphasize why we're doing research on scleroderma, because I think it's important to know that. It's important for you to know why, but it's important for you to gain that information so you can use it in your advocacy efforts and when you talk to your congressmen and your representatives and people in the community to educate them about the importance of scleroderma research. So we do research because the great use of life is to spend it on something that will outlast it. So that's why scientists do what they do, because any discoveries from our research will be there benefiting others long after we are gone. And why fibrosis research? So fibrosis ends up being a complicated process that can affect a lot of organs, but also a lot of different cells in the body, a lot of different processes. And the research, what the research, if you can imagine it, provides is little pieces of answers. So each experiment we do in the lab gives us an answer. That answer contributes to one piece of the puzzle. And as we put the pieces together, that's why we get a better picture as to what's happening in scleroderma or other diseases. And why specifically research on fibrosis? Well, we know that fibrosis can affect nearly any organ in scleroderma, but it affects, as you will see, organs and other diseases as well. Fibrosis is a leading cause of death right now from a variety of conditions, and we still don't have an FDA-approved drug that can actually reverse fibrosis. From a study from an investigator at the NIH, Dr. Thomas Wynn, had shown that fibrosis is actually responsible for nearly 45% of all deaths in the developed world. And you may think that's a really surprising high number. Well, there's a reason for that. Here is an example or a sampler of conditions where there is fibrosis. So let me see if pointing this way you can see. Can you see my mouse there? Maybe not. I don't think I have a laser pointer, but let's try and look at it. So if you look, for example, at the liver, fibrosis of the liver can happen for a variety of reasons. It could be hepatitis infection. It could be alcohol-related. Could be autoimmune, primary biliary cirrhosis. So there's a lot of reasons why you can get fibrosis of the liver. In the heart, very common conditions that you hear about all the time cause fibrosis of the heart. Things like congestive heart failure, like a myocardial infarction, where you get the scarring and the fibrosis in the heart. In the skin, it's not just scleroderma that causes fibrosis, but there are other conditions as well that have fibrosis in them. In the kidney, if you're diabetic nephropathy is kidney fibrosis. If you have lupus nephritis has a component that's fibrosis. So, and a variety of other conditions. In the lung, there are a lot of conditions. Who has not heard of someone who has asthma, for example? Asthma is fibrosis around the airways. Idiopathic pulmonary fibrosis, even pulmonary hypertension has fibrosis around the vessels. So a lot of conditions can cause fibrosis of the lung. In the eyes, you can have fibrosis in the eyes, for example, if you have glaucoma. So fibrosis is not unique to scleroderma. But what makes scleroderma special? Scleroderma patients have fibrosis in more than one organ: the skin, the lungs, the heart, the kidneys, the GI tract. And so if we can solve the puzzle of fibrosis in scleroderma, we can help the other diseases as well. So we call scleroderma a prototypic disease, and working on fibrosis in scleroderma and developing therapies in scleroderma will potentially help all patients with all the other conditions as well. So that's sort of the lesson, take-home message number one. Why do you explain to your congressmen and representatives why is fibrosis important? Why is research on scleroderma important? All right. So let's start taking a tour of the lab of how we do fibrosis research and other research. So this is a typical medical research lab. And I'm going to start the tour for our lab, where we are, with starting as you are entering into the elevators. Okay, we're going to start from the beginning. So this is how the elevator doors.
Ninth floor.
So here's the lab. You can't really hear the sound on this now, but so this type of lab is the model that's called an open lab space. In other words, when you get in and you see the lab, what you're seeing is a lot of research benches, those long things that have shelves where people do experiments, but it's a large space that has multiple of these where different people doing research on different diseases can be in one space interacting with each other and helping each other in that kind of setting. All right. I realized we practiced earlier and the sound came up on this and you could hear it, but for some reason I don't hear it anymore. All right. So once you get to the lab, this is the door as you go into the lab area. And you walk into the lab and it's what we work on are benches. These are benches. And your reagents, your chemicals, your buffers are on these shelves. And everybody works on a bench and has a little desk area to sit and work on the desk area. But then there are specialized rooms that have other things. So, for example, as you walk in, these are freezers where we store samples. And these little units that are called R2D2s are very specialized freezers. So our freezers go from your standard freezer that you have at home, which is usually minus 20 degrees Celsius, to the ones, there we go, to the ones that are a little larger that you're going to see here. Those are called, they have a temperature of minus 80 degrees Celsius. So we store in them things that we don't want to warm up at all or go through freeze-thaw cycles because that ruins some of the reagents we have. So as you go in, you can see across through the lab, you start seeing specialized rooms left and right. And as you go in here, the first freezer you will see on the white one is the typical one you have in the house. These tall ones are minus 80 degrees Celsius, and the R2D2 units are minus 140 degrees Celsius, and it's liquid nitrogen that cools them. What's unique about these is if you've contributed a punch biopsy or blood to research, who has done that for research purposes here? So when you do a little punch biopsy, we grow millions and millions and millions of cells out of that very tiny punch biopsy. And we can freeze down the cells in these R2D2 units. We can freeze them down for years and years and years. And we can pull them out and grow them again and let them multiply and give us even more cells to do experiments. So the gift that you give in a tiny punch biopsy could impact the research of several people for years and years to come. All right. So how do we do research? Initially, we do the research in the bench, which leads to discoveries, and it's those discoveries who lead to the therapies. The discoveries lead to the therapies, and the therapies are ultimately taken to the market by the larger pharmaceutical companies. And you start by asking a question. So you come up with a hypothesis, but you also do your homework. You go and see what's been published in the scientific field on that topic before, what's been done before, what has not been done, how can I ask a good question and design experiments so I can have good answers out of it. Then you design your experiments, you do the experiments to test your hypothesis, you analyze your data, and then you determine were you able to support your hypothesis or not. And ultimately, what you want to do is publish your results so other people learn from them, and so other scientists don't end up reinventing the wheel and repeating what you did. The type of research that's done is the top is what we do in a typical research lab. So it's for discovery purposes. So you make an observation and you're trying to come up with new observations, new discoveries. Ultimately, you want to test them in different settings. In vitro, which is in the purple, means outside the body. So that's when we grow the cells out of your punch biopsy or get blood out of you and get your white blood cells and do experiments on them. In vivo is inside the body, and usually that's when we use mice or other animals to test things. And ultimately, what you're trying to develop, let's say, is a therapy. But before you can get it to the patients, you have to test it. Is it safe? Is it toxic in any way to the patients? What is the half-life of the drug? How long does it last in your blood? How often do you have to give it? Where does it go? If you give something via injection, let's say intravenously, where is it going in your body? Is it getting to your lungs? Is it getting to your kidneys? Is it going to the liver? Is it getting cleared really quickly? So you want to know all those different factors. And sometimes you get to testing for safety, toxicity, and you realize, okay, well, this drug is not as safe as I'd like it to be. So you go back to the drawing board and go to the section in green where you have to now modify your drug and come back through the testing. And that's why it takes years and years and years to develop a therapies, because you may be going through that circle a few times before you have something that works well but is also safe. So the first part that is going to be there, the top part, the discovery phase of it, how do we do it? We take samples, so skin punch biopsy, blood, anything else from our patients who are willing to support the research and donate. And we do the testing that we're going to do, like we did with one of the molecules we looked at that we call here IGF-BP5. So you test in vitro, and as I mentioned, in vitro is outside the body. So the cells that we grow out of the skin punch biopsy are called fibroblasts, and that's what they look like under the microscope when we test them. They're these very long, elongated cells. You test in vivo, so in a mouse skin or mouse lung to know if this is going to work for skin fibrosis or lung fibrosis. And ultimately, there are other ways of testing that are more novel that I'm going to show you how we do them. So this is what, in the middle is what those cells look like. And what we do is we hit them with proteins or factors that can cause fibrosis, but then we also add in the mix things that we know can block fibrosis or can reverse fibrosis, and then you test, well, how much of this does it take to stop the fibrosis? How often do I have to add it? So a lot of different conditions that you have to test. And we can check different things. We can check as readouts, basically proteins. We could check RNA levels. We can check how quickly the cells are growing. Are they multiplying, not multiplying? So you can check a lot of parameters at that point. So this is what they look like. When we first start them out of skin punch biopsies or out of lung tissues, they look like the first on the left. So you see these long spindle-shaped cells under a microscope. And then when they start growing really fast, they stick together and become like a carpet, like a whole layer of cells, basically, and millions of cells. And what you can do is what we call expand them. So you will take cells that are growing in one plastic dish, and now you divvy them up into 4 dishes, and they fill the 4 dishes. You divvy them up into 8 dishes, and they fill the 8 dishes, and you keep growing them and have a fairly good supply for experiments. So how do we grow them? They were used in the body to a certain body temperature in certain conditions, and for us to do the experiments to sort of mimic what the cells felt like in the body, we have to maintain those conditions. So what you are seeing next to the fridge and freezer is what we call cell culture incubators. So these are chambers where we maintain body temperature, 37 degrees Celsius, and a little bit of humidity to be able to grow the cells. And we grow the cells in these little dishes. So let's see how this is going to go. And they're grown on shelves in these plastic dishes, and we keep them in a liquid that has nutrients for them that will appear pink now. So you can see them on the shelves. You can see the different types of sizes of dishes, and you can see this pink medium or liquid that they're in that gives them all the amino acids and all the nutrients that they need to grow. And to manipulate them and do experiments with them, we use these biosafety cabinets. You put a chair in front of it, and you do all the experiments inside because that's a clean environment. Because these cells, we have spores in the environment, there's bacteria on your skin, and all that can easily contaminate the cells, and then you have to throw them out. You can't use them in experiments. So we use what we call biosafety cabinets to be able to manipulate them, do experiments with them in an environment that keeps them clean. And this is what a cabinet looks like. You can see the purple lighting inside, that's a UV light. What happens is you turn on the UV light in advance while it's closed, and the UV light will kill any germs that are in there so that you don't contaminate your cells. And then you turn off the UV light so it's safe for someone to basically put their hands in and do experiments. And this is what it looks like when you're working inside a biosafety cabinet. You can manipulate small volumes. You can divvy up the cells like I talked, make more of them grow, all sorts of manipulations that you can do inside of that. And you can have two people working side by side if you have two of these at once as well, which helps facilitate the pace of the research and be able to do things a little faster. All right, so what does a readout look like from these cells when you do the experiments in them? Well, one of the possible readouts is looking at proteins. So who can give me an example of a protein that's made in excess in scleroderma? Collagen is the most common one. There are a lot of other proteins that are also made in excess, like collagen, one being fibronectin and others. But we can measure that. We can measure how much these cells are producing, because what they do is they make it inside the cell, but they secrete it out. So they sort of spit it out into that pink medium I showed you they grow in, and we can collect that and analyze it. So here's a situation where a member of the lab is collecting the liquid to analyze it. And we analyze it on a machine that's a digital imager like this. The screen of the machine shows you the proteins, and I'm going to show you what it looks like up close. So when you're looking up close, it looks like this. And you can see the darker bands. I know this is going to affect the video a little bit, but without a laser pointer, it's easier for me to walk. So these. So you can see there are some really dark ones that are very dense and lighter ones. So the darker it is and the thicker the band, the more there is of this protein. So you can see one, I put the name as collagen type 1. And the less there is and the thinner the band, the less there is of the protein. So if in the lab we experiment with the cells and we add something to them that makes them make a lot of collagen, this is what you get. If we add to them something that makes them reduce how much collagen they make, this is what you get. So that's how you test things that are going to help you reduce how much collagen is being made by the cells in the lab. And that, interestingly enough, is called a Western blot. So that's one of the assays that we talk about. All right. We have taken things in my lab a step further because, for one main reason, testing in the cells is great, but the cells are outside the body. And ultimately, there are other things in your body that affect how the cells behave and how the cells respond. Testing as animals is great, too. It helps. I don't particularly like doing that, but in research, it's almost a must to do that. But again, we are not mice. We're not going to behave like mice. And actually, most drugs that work beautifully in mice fail in human trials because it's different systems, right? Different things can be happening. And I can guarantee you that most researchers can cure just about anything in mice. The problem is that doesn't always then apply to the humans when you try and translate it in clinical trials for human conditions. So we've developed another way of testing things that involves human skin. And I apologize if anybody is uncomfortable seeing these images, but just to show you what it's really like in real life. So we get skin from, for example, tummy tuck surgery. So this is extra skin from plastic surgery that they're going to throw out anyway. What you see here is the top part is the skin, and the lower part is the fat. So this very thin layer, that's the skin. This is the fat. And we cut out the fat, and then we use the skin. And the reason being is we want to know if a therapy we're developing is going to work in humans. So we want to test it in a human tissue. And until you get FDA approval to do a clinical trial with humans and give the therapy to a human, this is as close as you could probably get. So this is what it looks like when you're working in these biosafety hoods with it. And then when you have trimmed the fat away, so you can see now the fat is on top and the layer of skin is on the bottom. You trim the fat away, and then you could use just the pieces of skin for our research purposes. So we developed a way where you could take human skin and you can actually cause it to have fibrosis. You can make it make more collagen, just like Scleroderma skin. And then we could add reagents to it, potential therapies, potential drugs we're developing to then make the fibrosis and the collagen go back down to normal levels. And what we do is we cut it up and put it in this pink liquid in these plastic dishes to let it have nutrients. And we keep the top layer of the skin or the epidermis exposed to the air, and we allow the rest of the skin to be exposed to the nutrients and the liquid so it stays moist. And that's how we use it experimentally in the lab. And one of the things we do is you see all these tubes. These have basically alcohol in them, ethanol. And what we do is we all have bacteria on the surface of our skin. That's normal. That's part of the normal microbiota of the skin. But for research purposes, that's not a good thing, because when you put the skin that has bacteria on it in a 37-degree warm environment like the body, all these are going to grow. And when you put it in a pink liquid that has sugars in it, nutrients, amino acids, the bacteria are going to grow and the fungus is going to grow. So we dip it in the ethanol quickly to first kill those off, and then we put it in the pink liquid. Okay, so I know you can't hear the sound, but what I'll do is I'll guide you over it. This is actually my lab manager, who's a very talented, very hardworking individual.
One of the
And
assays
he
we
is
do
explaining
is
in
a
this basically
human
what we
skin
do.
ex
So what
vivo
you see
culture.
is...
What we do for that is obtain tissue from surgeries like a tummy tuck or a breast reduction, and then we clean the tissue off, remove all the fat, dip it through a series of sterilizing solutions. What we end up with is a piece of skin that looks like this. From there, we can either do subdermal injections, or what we've been doing more
the
recently
eraser that's
is
on the back of a
using
pencil.
3mm punch.
So
similar
that
to
metal
your
part is
clinic.
what you punch
that,
a tiny piece of
we just
skin
have to
out
apply
of. That's how
pressure.
big it is. And we do the same thing. He's punching. You can see
And
he's
use,
punching
twisting
down
motion,
to
similar to
get
like a
his
drill.
tiny circular piece that's usually about somewhere between 3 and 5 millimeters of skin out of it.
And then we can move our punch
And that
into
skin,
a
that
6-volt
little
dish,
punch, he's going to transfer to another dish. You
where
can
we
see
can
that in the
further
lab.
treat
And then he will
and
add the
control
pink
for,
liquid to it, and he will
swelling
add
and
factors that cause
other
the skin
things,
to
and
thicken
test
and get fibrosis.
inducers of fibrosis.
And in the other
and
dishes,
also
he'll repeat the
peptides
same
and drugs
thing,
that
but
can
now
reduce the
add
fibrosis.
in addition something that may reduce the fibrosis. And by having a lot of little dishes, you can test things that cause fibrosis with things that stop fibrosis at different concentrations, different amounts, from very little to a lot, to figure out what is the best concentration that's going to work, what is the best amount. And this is sort of what it looks like. So now if you take that skin and you put it in wax and you make a slice of it and look at the slice under the microscope. So I'm going to orient you. If you just look at the top, this layer here, I need to be taller. This layer here is the outside of the skin, the epidermis, and then as you go inward this way. So the blue line tells me how thick it is. How thick is the skin? So how fibrotic is it? So if you were to guess whether we caused fibrosis in skin A or skin B, which one would you choose to be fibrosis, A or B? Who says A?
I say A.
Who says B? Future lab recruits for sure. B is the fibrosis. You can see how much thicker it is. Just that blue line is showing you how much thicker it got. So we know now that we can cause fibrosis in human skin that we use in the lab experimentally. And now that helps us test potential drug candidates that might be useful for fibrosis and for scleroderma in the lab before we get to the human trials, to know that it's likely to work in a human tissue, not just in mice or not just in cells in the lab. We do the same thing now with lung. So lung fibrosis is an important problem in scleroderma, but lung fibrosis is also an important problem in other diseases, like idiopathic pulmonary fibrosis, for example. We talked about asthma, having airway fibrosis. So it's important to also develop a means of testing that something is not just going to work in skin, but also in lung, because a lot of scleroderma patients have both skin and lung fibrosis. So for that, we've devised something similar. So up in the corner, you see what a human lung looks like. This is actually a scleroderma patient lung. And we get them, so I can imagine what everybody's thinking. How do we get these, right? So patients who are going to get a lung transplant surgery, we get the old lungs basically to use for research because they're getting new healthy lungs in the process. So these are transplantation lungs. And what we do is we cut them up into small pieces like the skin. So you can see in this picture where this lab member is actually cutting with scissors, holding with tweezers, cutting with scissors little pieces. I don't know how hard it is to see in the back, but see these little pieces? Mm-hmm. So it's these little pieces that we treat to cause fibrosis, because you can do that in the lung as well. And we can test if something is going to help make the fibrosis better. So this is what they look like when you put them in a plastic dish. They're fairly small. They're about 3 millimeters. So very manageable. And you can take a little piece of lung and use it for a lot of different conditions. And then we can image it. So we can look at that piece of lung to tell what's happening inside it when we're done. And this is what the microscope looks like. And everything is done with digital images anymore. So the old-fashioned way used to slow us down, but this really speeds up things. So for example, here we're looking at cells called myofibroblasts. So when fibroblasts that are growing in the tissues get activated to make more collagen, they also make other proteins that give them the definition of being what we call myofibroblasts. And then here we may use the stain to identify them. So what you can see through the eyepiece of the microscope, this is a piece of lung. And the green is the shape of the cells that are myofibroblasts that are making too much collagen. So now we can look, in this lung, how many of the cells are there? Based on the intensity of the green, we can tell how much are they making of these proteins. So we can learn a lot from them. Here's another way of looking at the lung. So this is actually a section of the lung, looking at it from a little further back, if you will, that shows you air spaces and cells. So this is a section of the lung under the microscope. These are spaces. These are areas here, for example, of collagen and fibrosis. And all the darker purple dots, that's the cells that we can detect, the nuclei in the cells. So you can pick up. But then we can look at it in those ways. All right, so we go back as to where that gets us. So when we test things in our system, whether in cells, in animal models, in our human skin or human lung model, we are testing in a system before we get to the further testing that allows us to develop the drugs. And you sort of have to do these steps before you can take a drug further. But what are ways of discovery of drugs that can be developed for scleroderma? So there are 2 different ways. You can discover brand new drugs and take them through the whole process from step 1 all the way to the end. Or you can take drugs that have been approved for other conditions that make sense to use in scleroderma because we know they reduce collagen or we know they help with fibrosis. And that's called repurposing of drugs. And I'm going to show you an example of one of those. So if you are repurposing drugs for clinical trials, what happens? So one example of that is a study we did where we showed that patients with scleroderma have very, very high levels of estrogen. And the gentlemen in the audience who have scleroderma may say, well, what about us? Well, it turns out that the men with scleroderma have even higher levels of estrogen. So to see what estrogen might do, we added it to the human skin, right? We take human skin, we added the estrogen, and we said, okay, well, what is estrogen's role in fibrosis? So the red arrow is pointing to what estrogen does. So if you were to look at this untreated skin, and this is skin treated with estrogen, which one's thicker? The estrogen. So estrogen can cause fibrosis in the skin. And actually, when we saw this, we were surprised. So one of the things we did is go back and try and read what was published before. Has anybody noticed this before? And it turns out that many of the companies that were developing hormone replacement therapy for postmenopausal women had noticed that when they were testing it, the women would get thicker skin with more collagen, and when they stopped the hormone replacement therapy, the skin went back to normal. But we didn't know that. And then we started testing drugs, so that's the green arrow, that block the effects of estrogen, and we saw that the skin could go back to normal, and we could reduce the fibrosis. So in this case, there are a lot of drugs that are out there to block estrogen. There are drugs that are used in breast cancer treatment, for example. So in this case, it would make sense to repurpose an existing drug, test some of these estrogen-blocking drugs to see if they would work at improving fibrosis in scleroderma patients. So that's repurposing of a drug. There's also development of brand new drugs. So how many people are attending for the first time in the audience, may not have heard my talk before? Okay. So several of the labs across the world who work on scleroderma, there's not that many labs that work on scleroderma, unfortunately, but several of them have new potential drugs that they're developing. So there's quite a bit in the pipeline that is going to be developed eventually, and some of these will make it to clinical trials. So many are developing drugs, which means they have to start from the first step of testing it initially in cells, doing experiment cells, and through the whole process. But there's quite a few basically coming up. One of them is one we've been developing that I've spoken about before, because what's interesting about the one we're developing is it's actually a piece of collagen. So this is a type of collagen called collagen 18. What we found is this tiny piece here, E4, that's from the tail end of collagen, can improve fibrosis. So we've tested it in our cells, we've tested it in the human skin, we're testing it in the human lung, and we show it improves fibrosis. It reduces collagen, it reduces other features or hallmarks of fibrosis. And this is when we tested it in human skin. So blue line is skin that's not treated. Red line shows how thick skin can get. This is, again, human skin when you make it fibrotic, so when you cause fibrosis, in this case with a growth factor called TGF-beta that experimentally most labs use to trigger fibrosis in the lab. And then the blue line again at the end here is when we give this peptide that we call E4 that we're developing, and you can see the skin becomes thinner again compared to the red line. What was interesting to us is you could give it and stop the skin from getting thick, but if you let the skin get thick and then give it later as a therapy, not as a prevention, basically, which is here, we still get improvement in the skin fibrosis compared to the red line. So that gave us hope, because when scleroderma patients come to the clinic, it's a little too late to completely prevent fibrosis. You already have skin involvement, you might already have lung involvement, but if we can find something that can reverse the fibrosis, then that would be an appealing potential therapy. So where are we? I'm sorry, yes. Yes, sir.
And maybe I'm misunderstanding, but you said, I think you said that you find that people with fibrosis have an increase of estrogen. Is that correct?
In their blood.
In their blood, okay. So obviously it's more prevalent in women, and they take drugs to help increase estrogen. Is that correct? So could there be a correlation between those drugs that the women take and scleroderma?
Right.
Does that make sense?
Right. So you're talking about the hormone replacement therapies, right?
Yeah.
So women who take those develop slightly thicker skin, but the skin goes back to normal when they stop taking the drugs, healthy individuals. In scleroderma patients, we found that if they have high levels of estrogen, they have bad skin fibrosis. But they're not necessarily taking these drugs. Their body is making the estrogen. Because in the men who have scleroderma, they're not taking these drugs. But what happens is their bodies are converting. They have an enzyme that's converting testosterone to estrogen. And that's causing the increase of estrogen in their blood. So there's an enzyme that's very active that's converting the testosterone into estrogen that raises estrogen levels in the blood. Great question, though. So where are we with this drug that we're developing? So as you know, many of the drugs that are being developed are fairly expensive. They're expensive because it takes years of investment of funds to develop them. It takes a lot of money to do the clinical trials, and it takes a lot of money to produce them. So for us, we had to find another option that would make it more cost-effective to make this peptide. And through our collaborators who developed this way of doing it, the peptide is actually being made in plants. So this is the plant that it's being used. It's a very distant relative of the tobacco plant. But what's interesting is you can engineer these plants to make the peptide in the leaves. Here's what it looks to have the leaves all grown. You grow them in these trays that get inverted upside down to make sure you infuse them with an organism that can express the peptide in the leaves. They are grown under controlled conditions on these trays for large-scale production, which is where the process where we're at right now. The pink light is the growth light for them. This is very controlled temperature. The temperature does not vary by more than 2 degrees from bottom to top. And then what you do is you just collect the leaves and basically put them in a chopper that chops the leaves, and you can extract the peptide from it. The difference is, and this is an estimate, so we'll see at the end what the actual amount is, but it won't be very different. The difference is for our peptide, in our case, if we were to make 1 gram of it synthetically, the traditional way, it would cost about $25,000 for only 1 gram. And if you make it through this process, 1 gram costs $50. So it's a much more cost-effective way of making peptides. For example, plants are very efficient at being engineered to make small proteins or small peptides, so it's a good way to do things. So that is sort of where we are with the manufacturing of our peptide in the large-scale production phase before entering phase 1 clinical trials to assess the safety. All right. So what can you do in research to help us speed up the process? Because everybody has a role here, right? I'm the scientist, I do the experiments, I design them, I analyze the data, we do the research, but everybody can contribute to research in one way or another, and everybody can help accelerate research and support research one way or another. So I just want to very briefly basically empower you with the information that you can use to support research advances in scleroderma. All right, so there is this quote by John Richardson that says, when it comes to the future, there are 3 kinds of people: those who let it happen, those who make it happen, and those who wonder what happened. So I'm hoping most of you will want to be in the group who makes it happen. There are different ways you can make it happen. I'm going to talk about 3 ways, 3 main ways that I think are important. One is advocacy efforts. As the patients and the caregivers, your voices will be heard, but it's important to voice your support for scleroderma research. It's important to voice the need to have more funding for scleroderma research. It's important to voice the need to have more funding for NIH, more funding for foundations like the Scleroderma Foundation, more philanthropy for the foundation, funds to be used solely for research. So that's one fairly easy way where everybody can contribute, right? Everybody can send an email to their congressman representative. Everybody can make a phone call. Everybody can make an appointment, show up in that office and say, you need to support scleroderma research. You need to increase funding for scleroderma research because scleroderma is a prototypic disease. Fibrosis in scleroderma is similar to fibrosis and all these other diseases. And if we can make a difference in scleroderma, we can look at all these other diseases and make a difference in them as well. So we have broad impact of the influence that improvements and advances in scleroderma research can have. Another way is to directly support the research program at the Scleroderma Foundation. So September 15th is our annual deadline when investigators from across the country submit grant proposals that get reviewed based on the science by a panel of experts in the field who rank the grants based on scientific merit, and the best scoring, more scientifically meritorious grants get selected for funding. But there's a limited pool of funds for it. On average, we fund about only 7 grants per year. We usually get 30 to 40 grants per year, so 7 is not a lot, but that's as far as we can stretch the funds. There's also different types of grants. So normally what we give is 2 types of grants. We have what's called the Established Investigator Grant. These are grants that are given to individuals who are established in the fields. A lot of the world-renowned experts who are here giving talks during this conference. They get $150,000 total that's spent entirely in their lab on the research, $75,000 per year for 2 years. We also have what's called New Investigator Grants. So this is a way to launch the careers of the younger generation, because we want to do the research, but we want to ensure that we're mentoring the next generation of researchers to help them take off with their careers, but also spare them reinventing the wheel and doing everything we already did and have them just move forward at a faster pace. So these grants support these up-and-coming investigators. Same total amount, $150,000, but this time it's spread over 3 years, so $50,000 per year over 3 years. And we give a total of 7 of both of these grants, so 7 total grants, whether you're new or established. We have funded just through the Scleroderma Foundation over $26 million to date in these grants, and we developed a new grant that was offered over the past 3 years. It's called the Multi-Center Grant. It's a way of encouraging centers across the country to collaborate, open communication between them, exchange of ideas, working jointly on projects which accelerates the research further. These are $500,000 grants. We can't give many of those. We gave only one last year just because it's a lot of money for the Scleroderma Foundation. However, there are naming opportunities for grants. So with donations, you have the opportunity to name a grant for a loved one, for yourself, for your chapter, for anything you want. So that's one way to support research. So that's the number 2 approach. So total number of grants in 1 year last year that we gave out was $1.5 million. And the third way is to participate in research, donate blood for research grants, proposals, projects that are ongoing. Donate a punch biopsy when you can. As I mentioned, from a tiny, tiny 3-millimeter punch, the size of the back of a pencil, the eraser of a pencil, we can grow millions and millions and millions of cells that can be used for a large number of experiments. So it really helps us, because without this, it's very difficult for us to do the research that's directly relevant to scleroderma. All right, and I will leave you with one of my favorite quotes, another quote that you could potentially use when you're raising awareness about scleroderma and in your advocacy efforts, by Mary Woodard Lasker. For those who are not familiar with her, she was a very strong supporter of research, and because of her efforts, we have the NIH budget and the NIH funds to fund research. Under her advocacy efforts, the NIH budget had a tremendous increase in the funding. And she said, if you think research is expensive, then try disease. So I thank you for your attention. I'm happy to take any questions. I hope this just gave you an idea how we do the research. Obviously, most investigators would be more than happy to give you personal tours of their lab. We're all always excited to share how we do the research because we appreciate all you do and how you support us in doing the research, because we truly could not be doing it without you. So thank you for all you do to support the research. Right, so I'm going to repeat for the sake of the video. So you can participate by giving blood or skin punch biopsy. Where do you go? The major research centers will usually say if they're doing that type of research. So if they have the bench research that can use that. And what they do is when you go for your doctor's visit, they'll have information about it as a flyer, depending how they recruit patients for studies, or they may tell you about it verbally. And basically at your visit, you have the option to sign a consent form to say you agree to do this, and they can do the punch on site. It takes 5 minutes to do a punch biopsy. It takes 1 minute to draw blood, so it won't delay you much. The risks are obviously very minimal. We rarely see anything go bad with a punch biopsy these days. So minimal risk and carries a lot of value for the research. But yes, a lot of the major centers are doing research, and they will let you know about it either in advance or during your visit, or they might have information in the waiting area. Well, the Scleroderma Foundation has a great list of all the centers. So you can reach out to the center that's closest to you and say, are you doing the type of research that requires us to give blood? Or, we'd be happy to help in that way. Yes, sir.
I'm just wondering, do you know what the breakdown is between kind of public and private sources of funding for research? I know the NIH is the biggest player in that, but what's given in by philanthropy or pharmaceutical companies in relation to what government provides?
Right. So the government right now, the NIH, is the largest source of funding in general for all diseases. Scleroderma, unfortunately, doesn't have as much money allocated for it at the NIH. So as an example, let me give you as an example. Scleroderma, the incidence of scleroderma and that of multiple sclerosis are not that far off, right? They're both fairly rare diseases, if you will, compared to, let's say, diabetes or others. Yet there's 10 times as much money for multiple sclerosis at the NIH as there is for scleroderma. That's why advocacy is so important. Industry usually does not fund the research happening in the academic labs as is. Usually what they want you to do as an academic investigator is test their drug for them in your system, in your cells, in your models. So they're not funding the development of the new ideas and the new research that's happening in the academic centers. Foundations, the main funder of scleroderma research right now as a foundation is the Scleroderma Foundation. There are phases where we've looked, and the Scleroderma Foundation was giving out more new grants per year, specifically very focused, where the whole project is on scleroderma, than was the NIH. So the money, the grant, the total sum per grant is smaller from the foundation. The grants from the NIH are usually, in general, larger. But right now, the Scleroderma Foundation, the NIH are the main sources.
Just one quick follow-up. And what's the biggest driver of costs of research? Is it the people? Is it the materials?
It's a great question. Yes, the driver of cost of research. So what many people don't realize is when you work in an academic institution, medical center, university, as a scientist, you're responsible to pay your own salary, your own fringe benefits. You're supposed to pay the salary of everybody who works in the lab and their fringe benefits, and you pay the reagents, the supplies, the costs of the techniques, the tools, the equipment that you saw, the freezers, the weekly delivery of liquid nitrogen to keep the cells frozen, all that stuff. So we are responsible for bringing in 100% of personnel, reagents, equipment, costs that you need for the research. So it's all of it, really. If I were to divvy it up, I'd say personnel is probably the highest cost, because you need the manpower, you need the people to do the experiments, and you have to be able to support them at 100%. Because they dedicate very long hours to research. Research is not a 9-to-5 job. It's not a Monday through Friday job. It's very long hours, pretty much 7 days a week. Yes, sir, and then we'll go. Right, so the question, are there clinical studies with estrogen blockers to look at if they affect collagen? Not that I know of. We applied to the NIH maybe 3 years ago with a grant proposing to do a clinical trial with one of them, and our score was really good. We came very close, but we didn't get the funding, so we're going to try again. So we are trying to get the funding to do an initial trial with one of those estrogen blockers in scleroderma, because it makes sense. It's a lot faster to go that route than to start from scratch. Yes, ma'am.
Did I miss, did you say where you were as far as clinical trials on your peptides?
Right, where we are with our peptide clinical trials. So we have not started phase 1. We are producing the peptide in large-scale amounts because you want to have a large batch of it that you can make sure is very pure, very clean, check the purity, you check the toxicity, you check all that, and then that same batch gets used for phase 1 safety clinical trials. So we are hoping to be able to start phase 1 within a year.
And how would you participate?
So clinical trials in general will get listed on clinicaltrials.gov. So that would be the way to identify it. The phase 1 is usually a small number of individuals, so it's usually one place, but it's also a very short duration compared to phase 2 and 3 trials. And then phase 2, and hopefully if we get to 3, will end up being in more than one location, and those locations will be listed on clinicaltrials.gov. Good question. Yes?
Are you, as a patient, allowed to participate in your own clinical trials?
Wow, tough question. No, actually, not only will I not participate in my own clinical trials, but I cannot lead them either because that's a conflict of interest. So one of the well-respected experts in the field, in our case at MUSC, that would be Dr. Richard Silver, will be leading the clinical trial. Well, thank you very much for your time. Thank you for coming for the lab tour. And if you are ever in the Charleston area, reach out to me. I'll give you a tour in person.